Good morning, everyone. Welcome to Virtual Breakfast. Today is April 24th, 2025. My name is Teresa Crook. I am the MSU Extension Field Crop Agronomist Educator. I am located here at the Saginaw Valley Research and Extension Center. I cover field crops, and I am fortunate to have 50% of my position funded by the Michigan Soybean Committee. This morning, we will have Dr. Erin Hill, the Weed Science Diagnostician at the MSU Plant and Pest Diagnostic Lab, with us. She'll be talking about herbicide resistance and water hemp focus.
Thank you, Teresa, for that introduction. And thank you, everybody, for having me here today. As she said, my name is Erin Hill, and I am the weed science diagnostician in our diagnostic clinic on campus, which is called MSU Plant and Pest Diagnostics. I'm not going to spend much time talking about our lab because I've talked about that on previous virtual breakfasts, but this is the group of people that I work with there and all the types of services that we cover. Throughout the presentation, I have several links, and I have them as QR codes, so that when people are listening to this as a recording, they can go visit the links, and we will make sure that we drop these in the chat. So this first link that I have for you is just a link to the diagnostic clinic and all of the services that we offer. But today, I'm really excited to talk to you about water hemp and its herbicide resistance issues. So I'm taking a note out of Dr. Kurt Steinke's book from last week's virtual breakfast, and I'm starting with a quiz for you. I have three amaranth species here, A, B, and C. And I'm wondering if you can tell from this photo which one of these might be water hemp. So I'm going to give you just a couple minutes. But letter A here is actually water hemp. And so you can determine, if you had that right or not just by looking at these pictures. Not kind of fair because you didn't get to examine the plant in person. But understanding water hemp and the resistance issues really starts with being able to identify it. And my experience in the diagnostic lab over the last decade has shown me that amaranth species are very commonly misidentified. And it doesn't help that one of my students' favorite tools, which are the plant ID apps, are really not that proficient in identifying which amaranth species you have. So this is from the article where we evaluate those apps in the class, and this is for Picture This, an app that typically does very well. And you can see out of all the categories of plants that we tested against, seedling amaranth, they're correct less than 50% of the time. So that's not very helpful. It is helpful if you're not sure if it's an amaranth species or not, because there are some other similar species like the Atriplex species. But once you figure out that it's an amaranth, you're really going to have to trust yourself to figure out which one that it is. So to cover that quickly, we're gonna sort of revisit a fact sheet that Dr. Christy Sprague came out with over a decade ago. It still holds very true because the look of water hemp hasn't changed in the last 11 or 12 years or more than that. So you can visit this. It's a really great reference. It has lots of a chart and lots of pictures. It's called Keys to Identifying Palmer Amaranth from Other Species, and water hemp is one of those that is included. So instead of going through that whole document, though, we're just going to go through the questions that I ask myself when I'm looking at an amaranth plant. The first one that I always go to is the stem hairy? Because this really cuts the field and narrows down your search. So here we have an example of a stem, and you can see that there are hairs pretty much the full length of the stem. Hairs can be a little bit tricky because as amaranth species grow, some of the lower hairs start to fall off. So if you're really trying to determine if that stem has hairs or not, you want to look up towards the top of the plant where the newest growth is, because the hairs are still probably there and they haven't been brushed off. So if the stems are hairy, so if your answer to that is yes, then you can tell yourself, no, this is not water hemp. It's also not Palmer amaranth. But if it looks more like this picture where the stem is pretty glossy and you don't see really any hairs on here, even up at the top where the newest leaves are, then you know that you're dealing with either water hemp or Palmer amaranth. The second question that I ask myself is, how long are the petioles on this plant that has a smooth stem? Are they really long? And by really long, I'm talking about this picture here, where you can see that the petiole is longer than the leaf blade, or at least as long as the leaf blade if you were to fold it in half. If your answer to are the petioles really long is yes, then and there's no hairs in the stem, then you're probably looking at Palmer amaranth. If your answer is no, it has shorter petioles and it also has a smooth stem, then you're looking at water hemp. So here's a picture showing those leaves side by side. These are 6-week-old plants. Now, it's important when you're evaluating the length of the petioles, especially for Palmer amaranth, that you're looking at the most mature leaves. So you need to look further down. So hairs, look at the top of the plant, but for these petiole lengths, look at those that are fully expanded because they do kind of extend over time. The third question that I ask if I need to get to that point is, what does the shape of the leaf look like? If it looks like a kite, that's what I think it looks like, or a diamond, or maybe a rhombus if you prefer. If your answer to that is yes, then, and you had, no hairs on the stem, then you're probably looking at Palmer amaranth. Or perhaps you missed some of the hairs on the stem and you're looking at one of our other common pigweed species like Powell amaranth, smooth pigweed, or redroot pigweed. If your answer is no, it does not have diamond or kite-shaped leaves, but it has more elongated leaves, then you're looking at water hemp. This is the only amaranthus species that we commonly have in Michigan that has these more elongated leaves. Sometimes we call them lanceolate, which means they're sword-like. So they are much less likely to have that diamond-shaped or those sort of subtle points out to the side. Finally, if you're later in the season, hopefully you've already identified it by that point, but if you're later in the season and the plants are flowering or producing seed, then you need to ask yourself, are there plants that don't have pollen, or are there plants that don't have seed, meaning there are separate male and female plants? If your answer is yes, then this is also a good indicator that you have water hemp, especially if it's met all this other criteria. Palmer amaranth is another species that behaves this way. All of our other pigweed species that we commonly find in field crops have both pollen or anthers and seeds in the future on the same plant. So the male and female flowers are together. So those are the questions that I go through. Usually hopefully we don't get to the part where it's got seeds or flowers on it. But those are the things that you want to cover. You can visit that fact sheet. The other fact sheet that I wanted to bring to your attention, there's one from Kansas State University. This one is helpful if you have more detailed questions about the seed. So you need to look at the sepals, those structures covering the seed, or they have some really good descriptions of the seedlings. So this is another resource out there for you for identification of water hemp and the other pigweed species. So moving on to resistance in water hemp specifically, all of our resistance data can be found on the MSU Plant and Pest Diagnostics website. We have an interactive map there, and the QR code, and we'll put the link in the chat for that, is available. You can go click on your county and you can find out what resistance issues that we have confirmed in the lab. This map, though, is just showing you where we have confirmed resistance in water hemp. So it's showing you all these light green counties that are filled in. Those are counties where we have confirmed resistance. Now, you'll note that there's kind of this section in there between these two light green sections that's still dark green. The diagnostic lab is not going out and collecting water hemp species and screening for the whole state. These are only based on samples that are sent into the lab. So it is quite possible that there are resistance issues in these dark green counties that we just haven't had a sample come in yet. Okay, so going through the types of resistance that we've seen over time, we've been screening water hemp since about 2011, and we've seen kind of a wide variety of issues that have come through. We've had some plants come through that are only ALS resistant, and so we're talking about resistant to herbicides like Harmony, First Rate, and Classic. You can see those designated with the little A in the county where we confirmed that. We did have one population come in that was only resistant to glyphosate, and you can see that. But most of the samples that come in end up being resistant to both the ALS inhibiting herbicides and to glyphosate. So you can see nearly all of the counties that were highlighted in that light green color are now showing you a triangle indicating that we have confirmed a case there, at least one. We do have a couple counties where the combination for multiple resistance in water hemp is ALS plus triazine herbicide. So that's things like atrazine or Princep, which has simazine in it. So just a couple there. And then unfortunately, our latest find with this last screen in 2024, we did confirm multiple resistance to both glyphosate and 2,4-D, which we had not previously seen in Michigan, and that was in Sanilac County. And I'll give you a few more details on what that looked like in a minute. And we have identified or confirmed one population that is resistant to 3 different herbicide sites of action. So that would be the ALS herbicides, glyphosate, and also the PPO inhibitors, which would be things like Flexstar, Reflex, or Ultrablazer. And that was over in Ottawa County. So because 2,4-D resistance is new to the state of Michigan this year, I wanted to show you what that looks like when we spray the plants, because to me, this is the ultimate fake out. So the plants start to look like they're going to die, and then they kind of come back like zombies from the dead. So this picture is showing you on the ends, the untreated plants. And in the center to the left, we're showing you what the plants look like with, this is a very low rate of Enlist 2,4-D 2 days after treatment. So you can see that plant second from the left is curved over. The leaves are curled and cupped, and that's typical symptoms that we expect from an application of 2,4-D to most broadleaf species. But then you can see that same population, and this is our resistant population, one pot over, which is showing you 4 days after treatment. So just 2 days later, we see that the stems start to straighten up and you start to see normal growth is resuming. This slide here is showing you on the left the 1x field rate of Enlist. It is 2 pints per acre as that 1x rate. And so you can see we have our known susceptible plant on the far left. That plant is curved over and starting to be crunchy looking, very necrotic, just 9 days after treatment, whereas the resistance biotype has kind of an S shape to its curve, and it's starting to grow upwards again. To the other extreme, when we're doing dose response testing to look at the level of resistance, we have sprayed an 8x field rate. So this would be 16 pints per acre. This is not a legal application in the field, but we are able to do this for research purposes in the greenhouse. You can see 9 days after treatment, the known susceptible on the left is toast, and the resistant biotype is starting to resume green growth. So even at this very high rate, we are seeing resistance in this population. This is an overall view of sort of what the dose response looks like. So these rows are increasing in rate from left to right. And this is 9 days after treatment again. So you can see a lot of these curved stems and some cupped leaves, but they are all still green and continuing to grow. If you want to find out more about how this population might be resistant to 2,4-D, there is a paper that looked at this in a Nebraska population, and it looked at metabolic detoxification. So if you're really into the nitty-gritty details of how this plant is resistant, you can follow this link and go to the article in the Journal of Agriculture and Food Chemistry to read more. But you can see this is their diagram from that journal article. You can see that those plants are doing exactly what our plants are doing, where they start to look like they're dying, and then they resume normal growth here in their resistant population. If you want to see what's potentially coming down the pipeline, there is a nice article on growiwm.org. This one is specific to water hemp, and it is showing you that in the United States, we have now seen resistance to 7 different sites of action. And it lists those here down the side. So we've already mentioned the ALS inhibitors, group 2, the auxin mimics, group 4. Now, an important point I did not mention that I should have about our 2,4-D resistant population is that it was not resistant to dicamba. So just because it was resistant to 2,4-D did not infer that it was resistant to dicamba. Continuing on, though, with the sites of action where we've seen resistance in water hemp in the U.S., the photosystem 2 inhibitors like atrazine, we've already mentioned, Roundup or glyphosate, the PPO inhibitors, but some new ones that we have not yet seen here in Michigan would be the pre-emergence herbicides like dual harness and Zidua, which are the group 15 long fatty acid inhibitors. And then also our bleachers or our HPPD inhibitors like Callisto, Lotus, and Impact. So we haven't seen those at this point. But if you look across this, you can see the multiple resistant populations, and these dots are indicating what combination of these different site of action groups that we've found populations. The most disturbing, of course, is this one down here at the end where there was a population in Illinois that is resistant to all 7 of these site of action groups. These blue boxes now are showing you the resistance that we've seen in Michigan thus far. Fortunately for Michigan, we're usually behind the ball when it comes to resistance issues. So we kind of see what's happening to the south, and we prepare and make sure that we're screening and looking for those things. So why are we seeing so many issues with water hemp? That would be because this is a native species. So unlike Palmer amaranth, water hemp is actually native to the Midwest and other areas. And so the populations in the seed were already here to some extent. I mentioned that there are separate male and female plants. That means every time seeds are produced, it forces that genetic material to be exchanged between 2 plants. There's no selfing going on. So there's a lot of genetic variability. And when you pair that with a really high seed production potential for these plants, it's really kind of the poster child for potential resistance issues. How to get samples tested, I just have 2 slides left. This one is talking about the things that we can do here on campus. We do have some limited tissue testing that we may be able to do with Dr. Eric Patterson, our weed geneticist. I talked to him about this recently, and he is fairly confident we can look at ALS and glyphosate resistance in water hemp through tissue testing and possibly PPO inhibitors. Unfortunately, though, there aren't tissue tests available at this point for those other site of action groups. So not for 2,4-D, not for dicamba or the HPPD inhibitors or the triazines. That means that we're still very reliant on the bioassays using seed, which is what I do in the diagnostic lab. So every fall, I put out an article with information on how to collect seed and when to submit it. Every fall, it's going to be due near opening deer season day around November 15th so that we can test those populations from November through March. And just as a reminder, we need a minimum of 5 plants that have seed on them in order to do this testing. You can also visit our website to get some tips for where to find the seeds on the various types of plants that we test. Finally, I'm sure there will be a lot of questions about management of these resistant populations. That really falls in the realm of Drs. Christy Sprague and Erin Burns, and they have a fact sheet in the back of the weed guide that addresses this specifically, along with Palmer amaranth. Currently, it's on page 163. You can also find it on their website. So I believe that Christy and possibly Erin also are on the call. So if we have management questions, they are going to help me answer those. With that, I'd like to thank you for your time. I'll be sure to put my contact information in the chat.
Alrighty. There were a few questions that came into the chat, Erin. Couple of them most of them for you. And so I would probably go ahead and I will repeat a couple of them. The first one was from Phil, because some people cannot see your response when you chatted it back to him. So Phil asked, does the use of cover crops have an effect on these weeds?
So my answer is possibly, right? I'm going to give you a very gray area answer here. I mean, when we look at cover crop research overall, we see that cover crops do have the potential to suppress weeds, right? But that's very much determined by the species of cover crop you use, the timing of planting, the timing of termination, and the level of biomass that you are able to accrue. So it's not a solid yes across the lines. If you plant rye and you terminate it really early and you don't have much biomass there to provide a mat for suppression, you may not see the benefit. And so I won't give it a blanket yes, but there is an indication that it can be helpful depending on the situation.
All righty, thank you. And Mark asked, you mentioned that the 2,4-D resistance in water hemp is metabolic. Does that give you any clues about how this will spread or how quickly or to other areas of the state?
Yeah, so I wanted to clarify that we suspect, based on the behavior, that this is metabolic resistance in Michigan. We haven't confirmed that yet. but we're pulling that from the research that has previously been done in my conversations with Dr. Eric Patterson. He would probably be the best person to answer this question, so I don't want to mislead everybody. I would suspect that the transmission may be a bit slower than if we have something that's controlled by a single alteration of the DNA. But I don't want to mislead everybody. I mean, we're seeing that this has appeared in multiple states in Michigan. And so we are not an isolated case.
I don't know if that's good news or bad news.
It's here. We need to be looking for it and we need to be screening for it. And you shouldn't be increasing rates to try and deal with this problem, right? That would be illegal. Even if you were allowed to do that, this population I'm showing you is not going to help in this particular case.
Got it. Always be aware, right?
Yep.
One of the questions that came in that do pigweeds cross-pollinate across species?
Yes, so it is possible for the pigweeds to cross-pollinate between the species. I don't see a lot of evidence in my, you know, 15 years of working
Yeah.
with the pigweed species,
It's kind of
but
funny.
yes, it is possible.
Okay. This is a follow-up, does the resistance follow that way then too when they cross-pollinate?
I haven't seen evidence of that in Michigan. That's not to say that it can't happen. So again, I'm going to give you a gray answer. So I would say theoretically that's possible, but we
Hey,
haven't
gotcha.
seen that happening, right? We're not seeing glyphosate resistance, for example, in Powell amaranth or in red root pigweed. And I haven't seen hybrids of these two species that are showing that type of resistance. That's just one example.
Got it. And then there was one more question for you that said, how fast can weeds develop resistance to different classes of herbicides?
Well, the thing is that sometimes it's based on selection pressure and genetic variability. It's sometimes the resistance issue may already be there before we implement a new tool. So it can be very quick. Usually we see a lag time in some of these other states by a couple years, but that's not always the case, right? Just given the millions, billions of seeds that are out there that are all variable, it's potential that resistance already exists. So the speed really depends on the situation. Lots of not
Okay.
gray answers for you today.
I was recognizing that, yes. One more quick question that just came in. Is it possible to moldboard plow the seeds deep enough to stop them from growing as seeds live for obviously 7 years or longer?
So we have seen success with that with our mare's tail resistant populations, and Christy might be able to comment on that a little bit more. So you may have some success with that in the short term, and then how long that lasts, depends on if you have any other inputs of those seeds coming in. And yes, they can live. I believe this pigweed species have been documented to live for at least 30 years in the soil profile. So that's something to think about.
Okay, a couple more questions keep coming in. On the fun question that you started with your slides, what were the weeds in B and C?
Right. So weed B was Palmer amaranth. So I can go back to that in a minute if anyone's interested. You could see that the older leaves had the really long petioles there. And then plant C was Powell amaranth.
Got it. Another question, any resistant to glufosinate, which I believe is Liberty?
Yes, as far as I know, not at this time, right? So I was just going through this yesterday and looking at the research that was compiled by Grow IWM and also looking at the international database. And so thus far, I do not believe we've seen resistance in this species to glufosinate. But Chrissy or Erin can correct me if I'm wrong.
While there's not resistance to water hemp with glufosinate, there has been reports of it to Palmer. And Palmer, amaranth, and water hemp are very closely related species, so it's just one thing that we're definitely keeping an eye on.
And I will mention to follow up on that, when we're doing these resistance screens and people are suspicious of resistance to, let's say, 2,4-D in this case, we're actually screening like 7 different site of action groups. Glufosinate is always included. So we are looking for that even if you don't suspect it to be an issue yet, so that when we come back to you with the results, we can show you what we know will not work because there's resistance and what probably will still work because we noted it to be susceptible.
Okay, they just keep coming, so I'll just keep saying them. So for Eric asked for those see and spray programs and other scenarios with taller weeds, what are the best options for controlling mid-season water hemp or palmer?
And that I will defer to Christy since she's here.
Yeah, thanks, Chris. Thank you.
So depending on what, obviously the crop's going to make a difference, but I would say in general, some of the things that we've seen, particularly with water hemp, if you have an enlist soybean system, the combination of, and as Erin pointed out, that we're starting to see a little bit of 2,4-D resistance popping up in the state. So we've actually had better luck with a combination of 2,4-D and Liberty on just our normal populations that have probably worked the best from a post-emergent side if you do have the Enlist D3 soybeans. If you're growing, let's say, non-GMO soybeans, pretty much you're left with either using something like Flexstar or Cobra. And as Erin mentioned, we do have some PPO-resistant water hemp, and I think it's probably a little bit more widespread than what we've had samples sent into the diagnostic clinic. So that's one that we're always on the lookout for from just being cautious. So it's really important to get those pre's out to help eliminate some of those water hemp populations that we're trying to control post-emergence.
Okay, thanks, Christy. Another question, is growing resistance to herbicides an argument for increasing manual cultivation for weed control? Should we be dusting off the implements to run through particular bad fields with strong weed pressure or controlling them that way?
I would say yes, we're needing to rely on all the tools that we have in the toolbox, right? Because relying on herbicides alone, like we're seeing in these other places where they have resistance to 7 sites of action, it is not going to be the key to success, right? We need to think about our cultural practices, how we can increase the competitive abilities of our crops through spacing, through fertility, through mechanical management when necessary, and also what was mentioned through the use of cover crops too. These are all tools in our toolbox. We need to continue to think about those, maybe dust some of them off in certain situations. And then we also need to continue to look to the future and what other practices might be on the horizons. There are several places looking at alternative methods of control using electricity or laser weeders to see if that's going to end up playing a role in the future.
Got it. Thank you. Just when we thought those little things, the rusty things out in the field rows were not going to be used anymore, right? All right. I think that was all the questions for the weeds. I didn't see anything for Jeff at this point in time. And I'll probably open the door to other specialists because there was a specific question this morning about wheat. Are any observations on early season foliar diseases and thoughts on fungicide applications? And I'm assuming this is directed to Marty and or Dennis.
Hi, Teresa. Yeah, so Nicole actually sent me some photos in southern Michigan of some advancing powdery mildew in wheat. So yeah, I would get out, scout, and certainly a fungicide application to keep that powdery mildew in check could be warranted. And so if we haven't made that herbicide pass and that's still possible, tank mixing that fungicide in with the herbicide could be a good option. The question was on the economics too, and we typically see on average around about a 4 bushel benefit from that early fungicide application, that T1. It's really going to vary a lot depending on the field and disease pressure. Obviously, if there's pressure there, you're going to benefit more from that application or not. But on average, we're seeing about a 4 bushel benefit to that early application.
Okay, that was, as far as I could tell, that was all the questions that I saw. I think we will can possibly wrap it up this morning. The only other comment I was going to say is that there is a drone meeting coming up here at the research farm. I put the registration information in there in the chat if you're interested. It is next Tuesday, and hopefully people can come. Thanks. And if there's nothing else going on this morning, I wish everyone a very safe and timely planting season. Thank you for all attending, and we hope to see you next week. Think safety first. Talio.